So thank you very much for inviting me to speak here today. I think this is a very important topic, genetics of epilepsy. So I'm a fairly busy clinician at BC Children's Hospital, the pediatric neurologist there, and also neurogeneticist. But I do do additional research work in genetics of epilepsy. So today, I'm going to be going through some of the updates, really important updates and advances in genetics of epilepsy. Just in genetics itself, there's been a huge change since over the last 7 years since I last spoke and wrote some information. But I do think it's important to give an overview of genetics and then go through some of the different roles that genetics plays in epilepsy. What is the important implications for patients and families from that perspective? And then for most of the last half of the talk, we will be focusing on some of those advances and the implications for patients and families from that perspective. I actually don't mind if you want to interrupt and ask some questions throughout the talk, but also at the end. This shouldn't be even an hour talk, so I think that we'll have plenty of time. So just to start off with, what is genetics? We use that term a lot. It's typically defined as the study of hereditary or how traits are passed from the parent to the child. And what do we mean by traits? So these are specific features or characteristics in a person, and each person has a different combination of traits, which makes us unique. You can have different eye color as described as physical traits. Some people have this widow's peak here, or even just how the lobe of your ear is attached to the side can be a specific trait. Other important traits can be your height, your weight, your predisposition to disease, and your health. Often traits are due to a combination of genetic and environmental factors and how they interact with one another. For example, with your weight, often there's a genetic component to what your weight is going to be, but you can modify that with other environmental factors such as diet and exercise. We're going to be focusing on the genetic today. The gene is your basic unit of heredity, and it's made up of DNA, and your genes have a specific genetic code which are important for producing proteins, and proteins are needed for normal body function. This is just a slide of a cell showing you where the DNA is packaged in our bodies, the cells of our bodies. It's within the nucleus, and that's this dark stained structure here. So we looked at on a larger slide, the DNA is packaged into these chromosome structures, which I'm going to show in the next slide again. This is this DNA blown up in very large size, and the DNA which makes our genes. It's the composition of our genes. We have about 20,000 genes in the human genome, and all of that DNA together is what we've referred to. The term is genome, and you'll be hearing that a little bit later in the talk. This is a slide of human chromosomes. So if you get a blood test and you isolate the white cells from the blood, you stain them a certain way, you look under a microscope, and this is what our chromosomes are. So we have 22 pairs of regular chromosomes. Half of our chromosomes come from our mother, and half of our chromosomes come from our father, and thousands of genes are packaged on these chromosomes. So we have 2 copies of each gene. Half of our genes come from our mother, and half of our genes come from our father. And right down here, this is what we call our sex chromosomes, or 2 X chromosomes. This would be a female, and X and a Y would be a male. And this next slide just demonstrates again, we're just showing the sex chromosomes here. This is the mother with 2 X chromosomes, the dad with an X and a Y. They have a daughter and a son. So the mom for the daughter passes on one of her X chromosome, and the dad passes on the other. And for the son, the dad passes on the Y chromosome, and the mom passes on one of her X chromosomes. I'm not sure this will work anymore, but we can use the mouse. So the DNA, which makes up our genes, is composed of substances called bases. And we have different bases, A, T, C, and G. And there's a specific sequence to this that makes an amino acid, which is the building block of a protein. So this refers to the genetic code which determines the amino acid, which together produces our protein, which is required for normal body function. So now I'd like to talk about the role of genetics in epilepsy and what are the implications for families and patients from a genetic point of view. People looked back, have looked into the genetics of epilepsy for quite some time now. And one of the first studies that they were looking at were twins. So twins, some twins can have identical DNA. And what they were finding is that people with these twins that had identical DNA, that both were being affected with epilepsy. And this is an example back in the 1950s where Dr. Lennox, where both of these identical twins, Constance and Catherine, had childhood absence epilepsy. And what you're seeing below their pictures is their EEG, showing the particular EEG pattern we see of absence seizures, which is 3 per second spike and wave. People then went on look to relatives of individuals that had epilepsy, and they found that those relatives had a higher chance of developing seizures compared to the general population. So this is further studies showing that there is a genetic role in epilepsy. And then they went on to do linkage studies. And what linkage studies does is that you look at very large families with epilepsy. They look at the DNA of those that have epilepsy, and they look at the DNA of those that don't have epilepsy in these large families. And with sophisticated studies called linkage analysis, they were able to narrow down where that gene might be located. And that was actually what led to some of the first epilepsy gene discoveries, was by studying these large families. I'd now like to talk about what I divide up into the major genetic categories associated with epilepsy. The first one being the single gene. So a single gene disorders refer to where you have an abnormality in a single gene, a change, or what we sometimes call mutation, that results in epilepsy. And this is an example of how you might do that. So the example that we show here is that you're changing one of these bases. And what this does is that you can no longer produce the amino acid that's needed, and so you have a faulty protein, and this can result in disease. So this is an example of a single gene disorder that can cause epilepsy. There are certain characteristics that we see in single gene conditions. They can, that change in that gene can happen for the first time in the child, or it can be passed down from a parent, and in other words, inherited. If it's inherited, it can be through different ways, dominant, recessive, or X-linked. For the more common types of epilepsies that we're aware of, like childhood absence epilepsy or juvenile myoclonic epilepsy, it's rare that we see single gene disorders causing these ones. These are usually more complex, and I'll talk about that a little bit more. But there's many genes out there, and we'll be talking about that a little bit later too with the talk, that a single gene disorder can cause epilepsy, but they can often have other symptoms along with it. So epilepsy is a part of what they have in the single gene disorders. What is not just specific to epilepsy, but an important feature in single gene disorders is that you can have the same gene change or mutation in one family, and one family member can have it and have a very severe form of epilepsy, but another person could have a very mild form. So there's differences in how that epilepsy is expressed. They may even have different seizure types. What else we can see is that a person can inherit that affected gene and not actually develop epilepsy, and yet another family member in that family does. And another feature that we see is that more than one gene that's affected could give the same type of epilepsy. Now I'd like to talk about chromosomal disorders. And remember that you think back to that background. So that's where that slide that I show you, that's where all the DNA is packaged, or your genes are packaged onto those chromosomes. So Chromosome abnormalities can also cause epilepsy, and the problem there is that you're losing parts or gaining extra parts of the DNA within the chromosomes. This slide here is showing you an example where you get a loss here of this part of the chromosome that impacts on the production of the gene and the protein, and that can result in disease. So chromosome disorders can cause epilepsy because either the DNA has been lost or there's extra DNA there, and we call that deletions or duplications. Often this is a new change that happens for the first time with the child, and in that case, the recurrence risk of it happening again for a parent is low. However, sometimes it is passed on. In that case, if a parent has that same affected chromosome, then the risk of having it happen again would be 50% or 1 in 2 with each pregnancy. Because chromosomes can contain, if you have an abnormality there and you can affect more than one gene, you may see other medical problems besides epilepsy in these disorders. The complex disorders are very important in epilepsy because they represent a vast number. So individuals with childhood absence epilepsy, juvenile myoclonic, juvenile absence epilepsy, for example, often they're thought to have epilepsy based on a genetic basis, but it's thought to be complex. So there's probably multiple genes, multiple changes in those genes, and interaction with environment that influences your risk to develop the epilepsy. So the pattern of inheritance in these families is less clear. It's more complicated, and it's tougher to find the genetic cause in these situations. Mitochondrial, this is not an uncommon cause for epilepsy that I see at Children's Hospital. What am I referring to with mitochondrial disorders? Well, this is a picture of a cell, and the mitochondria are within the cell, and they're actually what provide energy to the cell. Mitochondria, though, are not only controlled by the DNA within the nucleus here, which is what we've been talking about, but they also have their own DNA. And so what I'm talking about right now is the kind of conditions where the DNA of the mitochondria is affected. There are some specific features we see here in mitochondrial DNA conditions. And what's important to know is that the mitochondrial DNA only comes from the mothers. So if a mother has abnormal mitochondrial DNA, theoretically, there's a 100% chance of passing that on for each child. However, what's specific about mitochondrial DNA problems is that the amount of abnormal DNA that's passed on is extremely variable. So some children may be affected and others may not. Because the mitochondria provides that energy, it's important for multiple parts of our body. So in mitochondrial disorders, what we can also see is that other body parts or other organs like the liver, for example, or the eyes may also be affected. What's really important to know is that symptoms can really be variable in mitochondrial DNA disorders. This is showing a pedigree. So this is a mother from a past generation. Here, these round dots are representing the mitochondria, white being normal, black being abnormal mitochondrial DNA. And so the mother, we get our mitochondrial DNA from the mom. So theoretically, she can pass on the abnormal mitochondria to all of her children, but it depends on how much is passed on. So these people with the black and the white lines are affected because they have a higher number of abnormal mitochondrial DNA, whereas her other two children are not affected. The other important thing to note is that her son, even though he's affected here, doesn't pass it on because he doesn't pass on the mitochondrial DNA. It comes from the moms. But this mom here, who's not affected, does have an affected child. And again, that's because there are differing amounts of mitochondrial DNA that can be passed on to the children. So often a question I get asked when I see individuals is, well, what is the risk to my family members? So if a parent or a child is epilepsy, what is the risk to other family members that they will develop epilepsy? The key thing is what is causing it. And in genetics, that's often some of the reasons why we search for it. So if the cause is known and it's an acquired brain injury, such as a stroke or a brain tumor or an infection or severe head trauma, then that risk is going to be low and not significantly increase. So for example, if a mom has epilepsy because she had a severe brain injury related to a head injury in a car accident, for example, it's unlikely that her children are going to be affected due to that. The risk is not significantly increased. But what if it's a genetic disorder? Well, the risk will depend on what the genetic cause is. I've already talked about the chromosome and the mitochondria. So often in chromosomal disorders, that change happens for the first time in the child. So in that instance, the risk is low. But if it was passed on for a parent, the risk would be 50% or 1 in 2. In the mitochondrial DNA conditions, theoretically, each child is potentially at risk, but it's so variable how much abnormal DNA can be passed on. So someone may have no symptoms, or someone may have mild symptoms, and someone may have more severe symptoms. I'd like to talk about the single gene in more detail. So in single gene conditions, remember we talked about those and how it might be affected. If you have a new change that's happened for the first time for that child, then that risk for those parents is low. And this is something we tend to see, these new changes or de novo changes in the more severe epilepsy conditions with a single gene cause. If it's not new, if it was inherited, then the risk really depends on what the inheritance pattern is. And there's either dominant, recessive, and we'll talk about X-linked recessive. So this is a diagram explaining dominant inheritance. So what do I mean by dominant inheritance? Well, this dad is affected. So for a dominant inheritance, remember we have 2 copies of each gene. One came from our mom, one came from our dad. This dad has one affected gene, so he's affected. He has a dominant condition. The mother does not, so she has 2 normal genes. The risk of having an affected child is 1 in 2 or 50% with each pregnancy. And so you can see here that 2 out of the 4 children are affected. In recessive inheritance, you don't have the condition if you have 1 affected gene. You need 2 copies to be affected. So in this situation, we see a dad who is a carrier. That means he's fine, but he has 1 affected gene. This blue one has been changed. And the mom has the same gene that's also affected. So the risk of them having a child that's affected with each pregnancy is 1 in 4 or 25%. What is also important to note, though, for 50% of the time, they'll also have a child that's a carrier. So they're fine, but there's a potential risk for their children to be affected if they partner with someone who has the same affected gene. For X-linked recessive, this is something specific to the X chromosome. So here we have a mom who has an affected gene on 1 of her X chromosomes. But remember, females have 2 X chromosomes, so she's fine because she has 2 X chromosomes, just 1 of her genes is affected. But the chance of having an affected son, for remember to have, when she has a boy, she's either going to pass on the affected X chromosome or the unaffected. So there's a 50% chance that 1 of her sons will be affected. What about when the cause, yes? Yeah. So the X chromosome, remember with the sex chromosome, the 2 X's is a female and an X and a Y is a male. We always pass on one of our chromosomes, right, when we're having a child. The father is either going to pass on his X or his Y. So if he passes on an X, it's going to be a girl, right? Because the mom can only pass on an X. If mom passes on the affected gene to the son, so the dad has passed on the Y gene, then the child is going to be affected. But it's a 50% chance that she'll pass on one of those to the son. Yeah, but the father doesn't pass on an X chromosome to a boy. He's only going to pass the Y chromosome because that's what determines that you're going to be a boy. Okay? Is that okay with everybody?
So if
The daughter, so in the X-linked recessive, I've simplified it a bit because there are X-linked dominant conditions. But for X-linked recessive, no. But this one is going to be a carrier. So she has a chance with her future pregnancies of having an affected son. Okay? No, and this dad is fine. And this mom is fine. But she carries an affected gene on her X chromosome. Well, what about when the cause is unknown? So here, we're, in many ways, we're referring to many of those epilepsy syndromes with a genetic basis, but it's thought to be more complex. So childhood absence epilepsy, juvenile myoclonic epilepsy, childhood juvenile absence epilepsy. Complex inheritance, remember we spoke about that. It's a combination of multiple genes that have variants and or environmental influences. So the risk here is harder to predict, and it's really based on past studies. So there's a little bit of inaccuracy to this, but they've looked at it in the past, and what they've determined that if you are a relative of someone that has epilepsy, a close relative, then, and it's thought to be more of this complex inheritance, then your risk is about 2 to 4 times higher than the general population. The general population can run anywhere from 0.6 to 1%. So estimate about 2 to 4% risk. If you look at a parent who has epilepsy and having a child, in general, it's thought that the risk is less than 1 in 10, so less than 10%. There are certain factors that seem to influence that risk. So in individuals that have generalized epilepsy, that risk might be a little bit higher, or if it started at a very young age. And moms, a little bit higher risk for having the child develop epilepsy versus the father having epilepsy. I'd now like to move on some of these important advances that have happened over the last few years. And by doing that, I'm going to be talking about 2 main technological advances and how they have changed our understanding of the genetics or the discoveries of genetics of epilepsy. The 2 ones we're going to talk about are chromosome microarray, or array comparative genomic hybridization, long, tricky word, and the other one, next generation sequencing. And feel free to stop me along the way if you have some questions. So what is array CGH? In simple ways, remember our chromosome test where you looked at the chromosomes under a microscope, you're looking for an abnormality? That's basically what we're doing at a much more sophisticated, higher resolution level. So it's looking for very small DNA losses or gains on those chromosomes. And how does it do it? Well, all of our DNA is represented on this array in these tiny little wells. And then what is done is you take patient DNA and label it red, and you mix it with green labeled normal control DNA. So if the patient had the same amount of DNA or chromosome material as the control, then the color in those wells should be yellow, right? You mix green and red together, should be yellow. But if there's too much DNA in the patient in a particular area, you're going to get a red spot. Okay, so that's extra DNA. And if there's loss of DNA in a certain area in that patient on one of those chromosomes, and a region on the chromosome, you're going to get a green spot. So it really allows you to identify copy gain or loss of DNA at a much higher resolution than what we did before here. Yeah. What is this?
You said if it's more red and green works better than yellow.
There is no blue. There's only green. It may, I don't know how it looks to you guys, but there's only red, yellow, and green. That's yeah, those are empty wells.
Oh.
Sorry. Okay. That should only be those 3 colors. Okay. So if we compare to what we did before, so this is our naked eye looking under a microscope, knowing what it's supposed to look like, finding small losses or gains. You could only do that to a certain resolution. This is showing you a loss of DNA on a region of chromosome 15, so the resolution is much higher. So that's a huge development and progress. And this is just showing again what this looks for. We call them copy number variants, and this is an example when you have a copy loss. So we're going to focus on this D here, region on chromosome. If you have a copy loss or a deletion, you're going to lose that D. And if you have extra or duplication, you're going to have an extra D there. So that's what this test identifies, at a much higher resolution than what we were able to do before. So what have we found using this test in individuals with epilepsy? Well, we've made a lot of diagnosis in individuals with epilepsy, at least, or up to 15%. And we've looked at individuals here at Children's Hospital where we've made a diagnosis and we find one in about 15% of the time. And the diagnosis is much higher if you're looking at individuals with more severe forms of epilepsy. So that's treatment-resistant epilepsy or other severe forms where their epilepsy in itself can impact on their development. Other individuals where we're finding it in is if they have epilepsy plus additional features. So say they also have an intellectual disability or developmental delay or autism or other medical problems. The yield is much lower though if you're looking in otherwise healthy individuals that have controlled juvenile myoclonic epilepsy, for example, or childhood absence epilepsy, normal development, controlled epilepsy. The yield is lower there. There are always limitations to these testing. So it's possible that you get in a large percentage, we get a negative test, but it doesn't rule out a genetic cause of the epilepsy, and that's important to realize. It's possible that there are smaller duplications or deletions, that's extra DNA or missing DNA that perhaps we're missing, or it's due to a different genetic cause that this test does not identify. Remember those single gene disorders where you're changing the code? This test does not identify that. It's not uncommon for me, I'd say at least 20% of the time that I get a result back where it says, we're not really sure what this means. And what does that mean? Well, they find an extra DNA or loss of DNA in a region that's not in the normal database, but it's also not in the affected database. So often then we go to check parents. Well, do one of the parents have it? And that can help us to explain what we found. But sometimes we're still left with a little bit of uncertainty. Because we're looking at a person's all of their DNA, this test also puts you at risk for finding something you're not looking for. So sometimes we find an abnormality in a breast cancer gene, for example. So that's something that, for me, because I look after children, I make sure that I'm always talking to parents about what are the potential problems of doing this test with incidental findings or findings of unclear significance. Having said that, I do think this is a very important test, and it is available in BC, and it should be offered or considered in individuals where they have epilepsy of unknown cause plus some additional features, like they have severe form of epilepsy or they have epilepsy plus additional features. But it's very important. Counseling is done about this test before they get it. And now I'd like to move on to next generation sequencing. So this is even more new. We talked about how genes are affected. You can lose the DNA or have extra DNA, or you can change the code. So in the past, what we would do is something called Sanger sequencing, where you're looking at the code. We know what the code is supposed to be, and so we're looking for errors or changes in that code. And this is showing you an example where we've got a change. It's supposed to be a T, and it's gone to a G, indicating that there's been a change here that could be affecting that gene and causing disease. The limitations with this test in the past is that you could only look one gene at a time. So the advances with next generation sequencing is that we can look at lots of DNA, thousands of genes all at once, very efficiently, very quickly, and the cost is getting less, as I'll show. So what next generation sequencing refers to is that you're doing massive parallel sequencing, lots of fragments of DNA all at once. And there's three main categories that we look at when we're talking about next generation sequencing. We can look at the entire genome, so that's all of your DNA. That's a huge amount, huge amount of DNA. Or you can look at the exome, and I'm going to show a next slide to help explain that. But basically, that's the part of the DNA that just is used for making the proteins. Turns out that's just a small percentage of our DNA. Or you can be targeted. That means I'm going to look at 5 epilepsy genes with this type of test. That's what we're referring to targeted. So what this slide is trying to show you is the differences between whole genome, exome, and targeted. It turns out that a lot of our DNA, we're not really sure what it does. It doesn't actually code for proteins. And this is an example, say, of a strand of DNA from a gene. It's the exons that code for the proteins, and the introns are not. So whole genome, when we do whole genome sequencing, we're looking at it all. So you get a huge amount of data. There's a lot of variants that we'll see, and it takes a lot of time and challenging to analyze. Whereas the exome just contains the coding part, the part that's making those proteins. And that's only 1%, so it's just a tiny little sliver of that apple, of the whole genome. No, that's just it's referring to the strand of the DNA, the 5'M and 3'N. And then targeted. Well, targeted is you're just taking little pieces of that exome. So for example, for me, I often ask to get funding to test for a panel that will look at 489 epilepsy genes, for example. This is another slide just running you through what whole exome sequencing is. So you collect some blood. Sometimes they can do it on saliva. You're taking the DNA from that blood. You're going to just grab those exomes, or these exons. So that's just the protein coding part of the DNA. And then you're going to sequence all of it. You're going to figure out the code of all of it and see if there are any changes there in a gene that could cause epilepsy, for example. Challenge is that there's about 20,000 variants that we see per individual when we do this. So this is not a straightforward thing to do. It's quite challenging to find the cause. But what's important and helpful is that with the exome, you're looking at all of the coding part of the DNA, and that's a large percentage of the disease-causing changes or mutations. So about 85% of the disease in single gene disorders is found when you look in this DNA. So what has been the impact on genetics and epilepsy? So on what you see on the side here, these are representing all single gene disorders that we know about, many different genetic conditions. This represents the percentages that are described, and ones that have the cause have been identified. So you can see we're not doing so well here. We haven't really identified the genetic cause of many in the late 80s. We're starting to get better and find the specific genetic cause, and then we see a huge explosion here in genetic discovery, gene discovery for conditions. And a lot of that is due to the advances in the technology that we just talked about, and that number goes even higher if we include 2013 and 2014. So the impact has been a rapid increase in gene discovery. And what has been the impact on epilepsy? Now there's at least 500 epilepsy genes described, so genes that are associated with epilepsy. People are getting diagnosed more. And so when you look at the studies that they're looking at, a lot of the literature, the medical literature, they're focusing on the severe epilepsy conditions or epilepsy plus, and anywhere from 10 to 50% of the time they get a diagnosis. What they're finding a lot, especially in those severe epilepsy conditions, it's those new changes. So these are healthy parents. They have a child with a severe form of epilepsy. And so in those cases, that's a change that happens for the first time in that gene for that child. But there's many different genes. It's not just one single gene that gives us the answer. And that's why these advances and this technology is really important.
Did you have a question? Sorry, okay. Of course, there are always the challenges that come with the positive. They're still learning a lot with this next generation sequencing. So again, a negative test, there are limitations to this test. If it's negative, it doesn't rule out that there's not a single gene problem or a change. Again, we have this issue with variants of unclear significance. And how do we approach those? Could this be the diagnosis or not? These tests allow you to find, like I said, one of the most important things with this test is gene discovery. But that's hard to prove that, especially in a clinical setting. If I'm ordering a test, if I get funding, for example, to order a test, it's hard to prove that that gene, which we don't know in the past causing disease, is causing the epilepsy in that child. So it's challenging to prove for the new cases, novel genes. Access to this test is still very limited in Canada. There's also ethical issues with it, and this needs to be discussed before you get the test. And the cost is not cheap, but it's really come down. And this graph just demonstrates that. So if you look back in 2007, costing you $10 million to sequence all of your DNA, now we're down to less than 10,000. And on a research basis, whole exome sequencing or exome sequencing, we're probably running around $1,000 per person. So this is going to be something for the future. We're just not quite there yet in the clinic. What has been the positive impact on patients and families? Well, more are getting diagnosed. So these are individuals that have gone for years, many tests, they're getting a diagnosis. It's really important, the earlier diagnosis for some conditions, especially if there's treatment implications. So there's not many out there, but some epilepsy conditions with a specific genetic cause will impact on your treatment and change your management. So it's really important to identify those ones, especially of early onset. If you have a specific genetic diagnosis, you can give more accurate counseling to families. What is the risk it's going to happen again? What is the prognosis for this child? It's a lot easier if you know the cause than if you don't. The other advantage, put an end to that ongoing sea of testing that we're doing with kids when we don't, I work with kids, when we don't have a specific cause. If you find it, maybe stop doing all those LPs, MRIs that we continue to do. In BC at least, having a specific diagnosis can really impact on the amount of support you're getting in the community too. So it can also be helpful from that point of view. But with the positive also comes the negative. That's why it's really important that you have counseling before you get this test. So in some instances, we're finding a disease or a genetic cause where there's no specific treatment. And that can be very devastating for a parent. There's also potential discrimination for future insurance. Again, we have the problem with incidental findings, and there's been a lot of discussion around that, how to approach that. So because we're looking in some of those tests that I talked about, we may find something we're not looking for, and how do we approach that? So it's very important, again, that genetic counseling is done before this test is performed. So what is the current recommendations? Well, chromosome microarray, remember we're looking for DNA loss or gain with this test, much higher resolution. This is available in BC. It's one of my highest yield in diagnosing patients, and I think it's important to consider if someone has epilepsy of unknown cause, but they have additional features. So either they have treatment-resistant epilepsy or a more severe form of epilepsy, or they have epilepsy plus additional features. Again, counseling needs to be performed before you do it. The next generation sequencing, well, very limited in BC right now, getting a little bit better in Ontario. Right now, if we want to do it, it has to be on a research basis. Now we are in the process of having that available here for BC Children's Hospital. We'll be looking at individuals that have epilepsy of unknown cause with onset 5 or younger. We will be doing next generation sequencing on them. It's just not quite available yet, but that will be happening for us. This test will be available in the future on a clinical basis. It's just not quite there yet. I do think it's important to consider in individuals that have unknown cause of epilepsy and a more severe form of epilepsy, or epilepsy plus additional features, or early onset epilepsy. It's helpful to know that their chromosome microarray is negative. Again, counseling really needs to be performed, though, before it's done. What would I like to see for the future? Standardized approach to genetic testing. So everyone has the opportunity, and then we can get the diagnosis, if it is genetic, as quickly as possible. So if there are interventions, that it happens as quick as possible, especially for the young. I'm hopeful that all of these genetic discoveries are going to lead to something, like the development of better treatment options. If you know what is actually causing something at the biological level, the hope is that you can do something about it at the biological level.
More standardized testing.
Yeah. I also like to see more work or information from the pharmacogenomics of epilepsy. So I brought this slide 7 years ago, and there hasn't been a lot of change here. But what this is doing, this study involves looking at your genetic makeup and your response to medication. So really what they're trying to do is to see if there's a connection so that you get the best medication. You avoid the ones that can cause side effects, and you get the ones that are going to be most likely to treat your epilepsy based on your genetic makeup. There has been some progress in that we know that individuals that have this particular allele and those at risk of having this are of Asian descent. No, this is a particular allele, so that's your genetic makeup that we can test for, okay? And so people of Asian descent have a higher chance of having this, and if you have that, you have a higher risk of having a severe drug reaction to Tegretol. So at Children's Hospital here, if we're considering using Tegretol, we'd screen for this if you're of Asian descent. So the aims of pharmacogenomics is to try and individualize and optimize your treatment based on your genetic makeup. And I would like to see a lot more come from that. To summarize today, genetic factors play a role in many epilepsy disorders, and I think the new technology has revolutionized and will continue to in terms of gene discovery and genetic testing in epilepsy. And I'm hopeful that these recent discoveries and further advances in diagnosis, and hopefully in the pharmacogenomics that I just mentioned, will lead to improved individualized treatment of epilepsy. Our ultimate aim would be better outcome and better life for individuals with epilepsy. Any questions?
HML and B star, the people who 8% or have a higher chance of risk of.
So they have a higher chance of having that particular allele. So that's just a genetic feature, okay? And so if they have that, then they're at higher risk of getting a drug reaction to Tegretol. So what we do is that we check individuals of Asian descent if we're considering putting them on Tegretol. If they take Tegretol and they have that genetic feature, then they're at higher risk of getting a severe drug reaction. Yes? If it is genetic, it's likely one, but it depends on what your, has your epilepsy been described to be, for example, if it was thought to be juvenile myoclonic epilepsy, then more often than not, those are complex inheritance. And that means multiple genes and or environmental influences. And they can have different seizure types like absence myoclonic and generalized tonic-clonic. Very few individuals will have it just due to one gene, problem with one gene. Yeah. I think there's been some more with the Tegretol, and there's more work on that. But
That's the main one that we're testing for, yeah. Yes?
If someone's over 18, and say they have more of a mild, moderate form of epilepsy, not severe, severe, they're not epilepsy plus, if they wanted to get some sort of genetic testing, how would they do that? Would they cover?
So that's a very good question. So I think to start with your neurologist or epileptologist to see if could you have a genetic condition that's not just your complex inheritance, because sometimes some of the juvenile myoclonic or even some of the childhood absence epilepsy can be more severe. And in some of those, you may want to consider doing more genetic testings because that's not typical. But I think it's important to go back to your neurologist, your epileptologist, and talk to them. Should genetic testing be considered a need? And if that person is uncomfortable about how to go about doing it, they can refer to someone who's used to doing those testing. And in children's, for example, it might be myself, or sometimes we have them to go to medical genetics. They may see a genetic counselor. Medical geneticists may request some additional tests too, and talk to them about genetic testing. Yes. Oh, sorry. Yeah.
So, I have a child who has a diagnosis, but it's not, I don't think they specifically looked for epilepsy or a condition that causes seizures. Would it be something that I might want to bring up with my neurologist about doing more genetic testing for seizures?
It depends on what was found. So if what was found is really consistent with the type of epilepsy in your child, it's not necessarily something that needs to be looked at in more detail. But it's probably a good idea to go back to your neurologist and chat about that. You wanted to ask something else?
No, I was just thinking if you're a woman who has epilepsy and you're thinking about finding or getting pregnant, there's no situations is it necessary to bring it up with the neurologist when she is done and requiring finance? Are there any other guidelines you give in some sort of situation?
Yeah, I think, yes. So there are certain situations where, that it'll happen to me and where I might be seeing a child, for example, who has unexplained epilepsy, and especially if it's more on the severe end. I always ask the parent, are they planning to have for their children? I'm going to have them go and see a genetic counselor and consider genetic testing. So if you're asking if the woman herself has epilepsy and she has questions about how her child might be affected, again, I would recommend talking to the neurologist about that. And if the neurologist is comfortable or the epileptologist is comfortable about what that risk might be, and remember, I talked about some things where it's quite straightforward or what that risk would be. If it's less clear, then referring to a genetic counselor and or a geneticist would be a good idea.